Yeah, that's perfectly fine. Good.
We will, yeah. Does this? I guess it works. I think we'll wait one or two more minutes and then we'll get started. Okay, well, why don't we begin? Hello, everyone, and welcome to our first event of 2024. My name is Professor Ann Karagosian, and it is my privilege as the director of UCLA's Promise Armenian Institute to welcome you to what I know will be a fascinating lecture entitled Genomic Analysis of an Ancient Armenian Population from the Artsakh, Nagorno-Karabakh region and other areas of Armenia. Implications for public health policy in the Homeland and the Diaspora, delivered by one of UCLA's foremost geneticists, Dr. Wayne Grody. Dr. Grody is a professor in the UCLA Departments of Pathology and Laboratory Medicine, Pediatrics, and Human Genetics, and also serves as co-director of the UCLA Familial Mediterranean Fever Program, the largest such clinic in the Western Hemisphere. And so he knows the Armenian people very well as a consequence. His lecture is based on research that was supported in part by our UCLA Promise Armenian Institute, as you will see, which is a truly remarkable study, especially given its focus on Artsakh, Nagorno-Karabakh, and all that has happened to the indigenous Armenians of that region since their project started. After Dr. Grody's lecture, we will have brief comments by his two research collaborators in this project, Dr. Yevgeny Sokorenko, who is a professor in the Department of Microbiology at the University of Washington School of Medicine in Seattle, and Dr. Sevak Avakian, the executive director of the Armenian Bone Marrow Registry of Yerevan, Armenia, and a co-founder with Dr. Sokorenko of the Armenia project. I am also pleased to be joined in our program today by our good friend, Salpi Akaragian, the president and founder of the Armenian International Medical Fund and a former nursing director here at UCLA Health. Salpi will be introducing our speakers in more detail shortly. I am also happy to note that our event today is co-sponsored by the UCLA Department of Pathology and Laboratory Medicine, the UCLA Institute for Society and Genetics, the Armenian International Medical Fund, the Armenian American Nurses Association, the Armenian American Medical Society, and Dr. Garni Bakhurdaryan, and the Armenian Medical International Committee with Dr. Viken Sipilyan, as well as the UCLA Armenian Students Association. So we are grateful for the support of all of our co-sponsors. And on behalf of our co-sponsors here at UCLA, I would like to acknowledge our presence on the traditional, ancestral, and unceded territory of the indigenous Gabrielino Tongva peoples. It is a privilege for our institution to acknowledge the history of the land on which we are established. Before we move on with our program and the introduction of our speakers, let me just mention a few procedural items. First, let me note that this event is being recorded for future viewing at our UCLA Promise Armenian Institute YouTube channel, and of course is visible now on Zoom and on YouTube. Second, for those of you in our audience, when the talks are concluded, please feel free to raise your hand, and if you have questions, and we will have a microphone brought to you for you to pose your question. For those of you watching via the Zoom webinar platform, please feel free to type your questions into the Q&A feature of the Zoom window, and we will try to get to as many questions as are practical. For everyone, please try to make sure your questions are as clear and succinct as possible. And for those on Zoom, one more thing, in the unlikely event that the Zoom drops for any reason, please just reconnect to the same link you were given originally, and it should bring you right back to our session. So now it gives me very great pleasure to introduce to you Dr. Salpi Akaragian, who has been actively engaged in many aspects of the Armenian Genomics project from its early days. As I mentioned, Salpi is the president and founder of the Armenian International Medical Fund and is a former nursing director in UCLA Health. She received her BS, MS, and Ph.D. degrees here at UCLA's School of Nursing and has worked over many years in pursuing healthcare reforms in Armenia, including setting up a primary healthcare clinic in the Lori region and facilitating the formation of the Armenian Nurses Association in Armenia. In 2003, Salpi co-founded the cochlear implant program at the Yerepuni Medical Center in Yerevan and has coordinated the work of Dr. Akira Ishiyama of the UCLA Division of Head and Neck Surgery in performing cochlear implant surgery in Armenia ever since, so more than 20 years. Through their team's efforts, over 200 children and young adults have joined the hearing world. More recently, Salpi has also been instrumental in working with burn victims in the aftermath of the Artsakh gas explosion from last fall, which occurred, as all of you know, as over 100,000 indigenous Armenians fled after the tragic loss of their ancestral homeland in Artsakh. So, Salpi, I will now turn this event over to you for a more detailed introduction of our distinguished speakers.
Do you hear me? Wonderful. Thank you, Anne, for the introduction. Good evening, dear colleagues and friends of the UCLA Promise Armenian Institute. It gives me great pleasure to introduce to you the main speaker of this evening, Dr. Wayne Grody, and two special guests, Dr. Seva Gavakian and Dr. Evgeny Sokorenko, who traveled to Los Angeles for this special event. Dr. Grody is a professor in the departments of Pathology and Laboratory Medicine, Pediatrics and Human Genetics, and Institute for Society and Genetics at UCLA School of Medicine. He's the founding director of the Molecular Diagnostic Laboratories and the Clinical Genomics Center within the UCLA Medical Center, one of the first such facilities in the country to offer DNA-based tests for diagnosis of a wide variety of genetic, infectious, and neoplastic diseases. He's also an attending physician in the Department of Pediatrics, specializing in the care of patients with or at risk for genetic disorders. He has been one of the primary developers of quality assurance and ethical guidelines for DNA-based genetic testing for a large number of governmental and professional agencies. Dr. Grody served for 5 years as founding chair of the Advisory Committee on the Genomic Medicine for the entire VA healthcare system, and is the past president of the American College of Medical Genetics. He was the expert witness for the American Civil Liberties union in the historic Supreme Court case that individual patenting of human genes. A particular interest has been in the diagnosis, treatment, and population genetics of familial Mediterranean fever, a recessive genetic disease specifically prevalent in individuals of Armenian descent. But I understand there are also in the other nations besides Armenians, right? As far as I think I understood, even in China, somebody from China came down with that. This has led to much involvement with the local and international Armenian community and the launch of the Armenia project, aimed at characterizing genetic risks and ancestry of the Armenian population to inform new efforts in public health and prevention. As a sidelight, Dr. Grody has been active in the film and television industries for many years. First as film critic for MD Magazine, as technical advisor and sometime writer for a number of feature films, TV movies, and television series such as Life Goes On, Chicago Hope, Law and Order, Heroes, and Grey's Anatomy, and 2 movies, Virgin River and Nutty Professor. So I think we have somebody in the audience who understands quite a bit about the movies and television. So we appreciate all your good work, Dr. Grody, in this area. Dr. Grody did his undergraduate work at Johns Hopkins University, received his MD and Ph.D. at Baylor College of Medicine, and completed residency and fellowship training at UCLA. Special guests. Dr. Seva Gavakian is from Yerevan, Armenia. He graduated from Yerevan State Medical University and completed his postgraduate studies in Moscow and Ukraine. Furthermore, he completed fellowship programs in the US and Canada. Soon after graduating, Dr. Avakian was appointed by the Minister of Health in Armenia as a director of the International Department. He was instrumental in arranging to send dozens of children and adults who suffered from the 1988 earthquake in Armenia. Dr. Avakian was also the Deputy Minister of Health and initiated and continues to initiate innovative healthcare projects for Armenia. Thank you, Seva. Currently, he is the Executive Director of the Armenian Relief Society's Akhurian Mother and Child Health Center in Shirak region in Armenia, and the Executive Director for the Armenian Bone Marrow Registry in Yerevan, Armenia. Most importantly, for this event, Dr. Avakian is the co-founder of Armenia, so we welcome you to UCLA and to Los Angeles. The next speaker, guest speaker, is also the co-founder of Armenia, Dr. Evgeny Sokorenko from Washington, Seattle, Washington. Dr. Sokorenko is a professor of Department of Microbiology at University of Washington School of Medicine, Seattle, Washington. He is a founding board member of the Armenia Project, ARMADA Foundation, and ID Genomics Incorporation. Dr. Sokorenko's interests are in genetics, evolution, and epidemic spread of human pathogens, population-wide susceptibility to infections and somatic diseases, vaccine, and monoclonal therapeutic design and development of rapid DNA-based diagnostic tests. He has pioneered several fundamental concepts in biomedical fields, including the pathoadaptive evolution, which I can't even say, right, of human pathogens, the catch-bond mechanism of protein receptor interactions, clonal diagnostics of infectious diseases, and others. Dr. Sokorenko and his colleagues' research was featured by Natural History magazine, National Geographic, and other magazines as well. He has also been featured on TV and radio for his scientific work. Dr. Sokorenko was born in Ukraine, received his degrees from the Moscow Medical Academy, and in 1990, we were lucky, he moved to the United States so he can continue his research career. Dr. Sokorenko and his wife, Dr. Arpenik Avakian, are the co-founders of Armenia Project. They support various Armenian medical foundations and continue to contribute to the cause of the Armenian people through different means. Can I get a hand of all these 3 wonderful people's biography that we read? So for now, it is with great pleasure and without delay that please help me welcome Dr. Grody to the podium to begin this evening's presentation titled Genomic Analysis of an Ancient Armenian Population from the Artsakh, Nagorno-Karabakh region and other areas of Armenia. Thank you.
Thank you, Salpi. Wonderful introduction. It's such an honor to be the keynote speaker tonight. She warned me she was going to mention that Hollywood consulting stuff, so it is a lot of fun. But I've been advised not to give up my day job, which is a little more secure. But just to give you an idea of how I prioritize things, I've told a couple of my colleagues there. I actually had an invitation to attend the Academy Award nominations announcement this morning in the Academy's headquarters in Beverly Hills, which I've never had. I've been in that building, but I've never been to that event. But I told them I can't do it. I've got a very important lecture later in the day, and I just can't. I've got to be sharp and not cloudy. As some of you know, in order to get the nominations news onto the morning shows in the East Coast, the event starts at 4 o'clock in the morning. So I would have had to get up really early, and this is much more important, believe me. So yeah, it really is an honor. I've been friends with Salpi for so long, and then more recently with Sevak and Evgeny. We are talking about the Artsakh region tonight, but I hope the findings that we will show you can be extended to the greater Armenian population, both in the homeland and, as the title says, in the diaspora here. So Sevak came prepared. He gave me, this is the Artsakh Nagorno-Karabakh flag, or I guess what it used to be. I guess that, you know, there is no such country anymore, or region. You know, as Salpi said, very sad. Don't worry, I know this is a diverse audience. I'm going to keep the technical details to a minimum here and try to keep everyone engaged and entertained. I wanted to begin with how did this start? Oops. Okay. Salpi mentioned it a little bit. How did I get involved with Armenian genetics and the Armenian people? You can tell from my last name, I'm not Armenian. I do belong to another ethnic minority that many people have said has much in common with the Armenian people in history, culture, love of food, things like that. And my friends know that I sometimes go on in more detail about that and make jokes about it. Unfortunately, as you all know, both groups are in a very fraught, difficult time now, as well as the third war that Evgeny represents here, Ukraine. So I think I'll probably leave it at that for now. But I have come to really love the Armenian people. And I've been there twice. I've been to these places, and I'll give you a little detail about that history. Of course, before that, I've been to Little Armenia, otherwise known as Glendale, and North Hollywood. And that's where I really got involved with local Armenians. And it really comes, as I'll give you in more detail, from the large number of patients with familial Mediterranean fever we have here in Los Angeles, since we have more people of Armenian descent in Los Angeles than anywhere else in the world except Armenia itself. And I've really come to be very involved with both the local Armenian population and in the homeland. And although I'm not Armenian myself, I've been assured that I can call myself an ABC, Armenian by choice. So it has been largely from seeing so many patients of Armenian descent with familial Mediterranean fever. Because of that, I've become kind of an expert at it. Salpi mentioned that it has a very high carrier rate in Armenian people. In fact, the highest carrier rate of any recessive genetic disease that I'm aware of. You can see here it's 1 in 5. As she said, it is found in other groups throughout the Middle East, Arabs, Jews, Iraqis, Italians, Greeks, and so on. but the carrier rate is highest in the Armenians, and therefore the incidence and prevalence of the disease is highest in them. People asked why is it so high, 1 in 5? Does it have some selective advantage? There are two reasons why mutations for a recessive disease might be especially high in a particular ethnic or racial group. One is called a founder effect. That's when the families who started that group thousands of years ago happen to have that mutation, and they've largely stayed isolated or not intermarried too much, and so it's just spread throughout that group. In Ashkenazi Jews, Tay-Sachs disease would be an example of that. There's no selective advantage to being a carrier of that, and the disease itself is horrible with early infant deaths. The other reason is a selective advantage. The example of that would be sickle cell anemia, which originated in Africa. It has a carrier rate of 1 in 10 in pure Africans, less in African Americans because most of them have some Caucasian blood because we know what happened on the plantations. But that has a highly selective evolutionary pressure. I think any of you who've been in the healthcare field know that although the sickle cell disease itself is horrible, especially in a third world country, it's fatal. The carriers are generally healthy, and in fact, they're relatively resistant to infection with the malaria parasite when they get bitten by the mosquito. So in evolution, that was seen to be a decent trade-off. Evolution has no emotions, and that it's totally just based on probabilities. And evolution, although it didn't think this out, it arrived at the conclusion that it's worth having a small number of children die a horrible death with sickle cell in order to have 10 times more people who are the carriers survive malaria. And that's how the mutation gets passed on. There have been some theories about FMF that the carriers might be relatively resistant to some pathogen. The one that's been talked about the most is Yersinia pestis, which is the bacterium that caused the Black Death in the Middle Ages, Black Plague. I have some doubts about that myself. I think it could just be a founder effect. Armenia is, bounded by mountains. It's always been isolated, and it is a group that tends to marry within themselves. As Helpy said, it can be found outside the Middle East. I have patients who are pure Chinese, pure Japanese. When we sequence their FMF gene, and I'm going to tell you a little about the gene in a couple minutes, strangely, they have the same mutations, the same nucleotide, the letters of the genetic code altered as we find in the Middle East and Armenia. The chance of that happening, there's 6 billion nucleotides in the human genome. The chance of lightning striking twice across the world in the same spot is very unlikely. So I think the mutations were brought there by somebody, maybe through the Silk Road, Marco Polo, whatever. That's for anthropologists who study human migrations to come to a conclusion for. And so I become kind of a default expert on this without actually really trying to. This was a lecture I gave in our pediatric grand rounds a few years ago, and the pediatric administrator found that photo on the internet. I don't know if she's really an FMF patient, but she's feeling miserable with the fever. And why talk about such a rare disease? Why did I get interested in it? It's very easy to treat. You just take a certain medicine every day, and it prevents the attacks. But the vast majority of physicians in this country have never heard of it, so it's not on their radar. The symptoms are very nonspecific, so when the patients show up in the ER, they just ascribe it to a virus or some other common thing and send them home. So many of them go undiagnosed or misdiagnosed for years or decades. Hopefully that shouldn't happen in the major cities like LA and New York, where we have a lot of people from the Middle East, but it does. It turns out that with the number of Armenians in Los Angeles, this probably represents the largest cohort of patients with a single rare disease, certainly on this campus, probably in California, and maybe actually anywhere, except maybe Armenia itself. So it's interesting for research. These are the basic symptoms. I won't go over all of them, but about once a month, they get a very high fever up to 104, 105, lasts about 2 to 4 days. There's nothing much you can do about it. They stay in bed. They typically have horrible abdominal pain, which is called peritonitis, and they can get arthritis and other things like that. At the end of the attack, which lasts, like I said, 2 to 4 days, it spontaneously disappears. They jump out of bed and they're fine until the next attack. The pain of the peritonitis is described as the worst anyone can imagine. It's kind of like what patients with appendicitis feel, maybe if the appendix had burst. I've had women tell me it's way worse than labor pain. On the pain scale of 1 to 10, they inevitably say it's 10 plus. And they're getting this every month for 2 to 3 days and missing work, missing school. No one believes you could be sick the same day every month, so they think they're just malingering and they get fired from their jobs. I've had patients who I've diagnosed in their 40s tell me that they had actually considered suicide because the thought of going through this every month was so horrible. Like some autoimmune diseases like lupus, strangely, the symptoms can kind of go away in females while they are pregnant. And I'll never forget one woman I diagnosed in her late 40s, had never been properly diagnosed before. She actually had 8 children because the only time she felt good was while she was pregnant. And it's sad because the cost of raising 8 children and putting them through college compared to the cost of this little bottle of the drug colchicine, which if you take it every day, it should prevent the attacks. It's, a huge difference. So, it sounds a little funny, but it's actually very, very sad. The gene was discovered in 1997 by a consortium of groups based in Israel, National Institutes of Health, and right down the street here at Cedars-Sinai. And that's how I first got involved with FMF. This was a research lab, and they had no interest in offering their discovery as an actual clinical DNA test. So they gave the cloned DNA to me, and I think we became the first center to actually do DNA testing for it. And that's when I started getting very involved. This is the requisition form of my lab. I've circled the FMF test there. And since we were offering the test since the mid-90s, I was invited to start attending our UCLA FMF clinic. This long predated my arrival at UCLA, and I've been here 40 years. It's at least 65 years old. As Salpi mentioned, I'm the co-director. Terry Getzug is the medical director. She's a gastroenterologist, meets once a month. It's in the Department of Medicine, so they won't see patients younger than 16. So I see all the pediatric cases myself in our medical genetics clinic. At UCLA, this division is called digestive diseases. Most places you'd call it Division of Gastroenterology. And in fact, in most other centers, if they see FMF patients at all, they would be seen by rheumatologists because it's an inflammatory disease, like rheumatoid arthritis. It's actually different, but I won't go into those details. But here it's always been in the GI division because the patients initially showed up with abdominal pain, and the original founders were 2 now deceased UCLA gastroenterologists. So that clinic is in the Division of Digestive Diseases, and the current division chief is Eric Israelian, who many of you know. He is the founder of the Promise Armenian Institute, but he's also very interested in our work on FMF, and he's a great partner. He's great at PR and raising the profile of this work. He does outreach to our local Armenian community here. I'm not sure if I'm allowed to show this, but he kind of likes to keep a low profile about that. So FMF is a genetic disease, and it's in the category of recessive. Autosomal means not one of the sex chromosomes, X and Y. And as shown here, if the two parents are carriers, they're healthy. Each one has a 1/2 chance of transmitting the mutant gene with each pregnancy. 1/2 times 1/2 is 1/4. So they have a 25% risk of having an affected child with each pregnancy. So this is called a Mendelian disease because it follows the laws worked out by Gregor Mendel. Now, Mendel was not Armenian. He was an Austrian monk, although he has something in common with Armenia because he did his work in the city of Brno, which was in Austria, but now it's in the Czech Republic. So with the various wars, these borders keep changing. And I actually was privileged to visit there in 2022 in the summer. I was invited to speak at the 200th birthday celebration for Gregor Mendel. So it was a real honor. This was held at the monastery in Brno, where he actually grew his pea plants in the garden. I think any of you who had, high school biology will remember he did the crosses using pea plants. And it was a real big deal for both Austria and the Czech Republic, and especially for the city of Brno. For about 2 years prior to the event, they actually had a giant pea that was making its way around the city. And I got to stand close to it. It's about 10 meters in diameter. And I had a private tour of the abbey, and those are some pea plants. I don't know if they're the direct descendants of his pea plants. There's also a really nice Mendel museum there, as well as a gift shop. And I'll just show you one thing I bought at the gift shop, which was kind of cute. So FMF and the other diseases we're going to talk about today follow Mendel's laws. Some of them are recessive, which means there's a 25% recurrence risk if you've already had one child with it. Others, such as the familial cancers, are dominant. And he worked this out with the pea plants also. A dominant disease, if you have an affected parent, there's a 50% chance that you inherited the mutant gene. So it's a 1/2 risk instead of 1/4. Of course, science has moved on in 200 years, and the same here at UCLA. We now do genome and exome. It's another kind of whole genome sequencing where we look at all 22,000 genes at once. We were actually the first center in the world, I think, to offer this as a clinical test. Instead of doing just one gene, one disease at a time, we do all 22,000. And it's reserved usually for patients with undiagnosed disorders. For this to happen, there had to be 3 advances first. One was the completion of the Human Genome Project, the sequencing of the first human genome, and that occurred in 2003. We need that because just like any experiment you do or any clinical lab test that's done in the hospital, you need a negative control. And the negative control is the reference genome. It's the one that was sequenced in the Genome Project. It's not exactly a healthy genome. There are no totally healthy genomes. We all have thousands of mutations, we now know. But over the years, we've gotten sort of a consensus reference genome. Then you needed a very efficient way to sequence all the letters of the genetic code. And that has been dubbed next generation sequencing. It came around in about 2011. The old way of sequencing, it took about 2 or 3 days to get a sequence of 100 nucleotides. I've told you that the whole genome is 6 billion nucleotides. So that was not even feasible at the time. Now with the current instruments, we can do 20 billion nucleotides in one run, several patients plus all the controls. Now the trouble is it produces a lot of big data and no one can manually go through all that. So the third criterion for doing this work was the advent of great computer power. If we sequence the genome of any of you in this room, the raw data takes up 5 terabytes of computer memory. It's really a lot to go through. So we're helpless without the computers. Unfortunately, I'm in the generation that didn't have computers until I was an adult, and I still don't totally trust them. I'm not very good at them, but I just have to rely on it. So I saw my own work at UCLA change almost overnight from molecular medicine, one gene, one disease at a time, to genomic medicine, all the diseases. So let's bring this back to Armenia. When the Armenian American Medical Society, which Ann already mentioned as a sponsor, had their meeting in Hollywood. It was, I don't know, 10 years ago or something. I gave a talk on this stuff that I just told you. And afterwards, Salpi, who I hadn't met yet, was in the audience and came up and introduced herself. You've already heard a little bit about the work she had already been doing on the cochlear implants. It's an amazing, wonderful success story. And this was done through her foundation, the Armenian International Medical Fund. At that time, they were thinking of branching off into some genetics or genomics. So she then introduced me to Sevak, who was also in the audience then, and it led to a memorandum of understanding, which he may talk about in more detail, that was signed at UCLA. You can see the bunch of us sitting there. In 2015, which was the 100th anniversary of the genocide, so it was very appropriate. We had seed money to start it through the generosity of the daughter of a genocide survivor, Sarah Chichian, shown here. And this is her signing my copy of her book, which she had written about her father, who was the genocide survivor. She's unfortunately now deceased, but we really need to give her credit for getting this whole Armenia project off the ground. This is one of the brochures that was made, which I didn't make up that tagline, but I like it, genetic mapping of the fractured homeland. And these were our goals from the beginning, to discover genetic variants that are specific to the Armenian population, to identify genetic markers that could be used maybe for screening for various genetic diseases, both recessive and dominant, and to have kind of some technology transfer, exchange of knowledge and techniques that could be used there. As you'll see, in Yerevan, they don't have all the fancy, really expensive equipment that we have here. So we need to adapt any tests we develop to them. And so a year after that, I went with Salpi and Evgeny to Armenia, gave some talks at the medical school there and other places in Yerevan, and then took the 6-hour drive over the hills to the region of Artsakh. I think most people here are very familiar with that already. It was a semi-autonomous region contested for decades with Azerbaijan. A very, very complex and most recently tragic history. You know the various wars, the one in 1990, which when they were still recovering from the earthquake, another terrible one in 2020, which was right at the height of the COVID epidemic, which had hit Armenia very hard. And these were launched by Azerbaijan, deliberately during those times. And then most recently, last year, a surprise attack that ended up conquering the area with the expulsion of, 150,000 or more people. So the region itself has always been 99+% Armenian Christians. I've never been sure why Azerbaijan wants it. I don't know there's any great resources there. I mean, it is an example of ethnic cleansing, if you want a good example. It is a complex history, though. And while I was there on both trips, Sevak and others gave me some nice history lessons. The best I could conclude that everybody agrees on is that somehow it was all Stalin's fault originally. It was some deal he made, in the 1930s that led to this continuing problem. But the one reason we're excited about this project, to our knowledge, there's never been any genetic studies of the Artsakh population. There have been in Armenia, although not the kind that I'm showing you. So this was taken on the 6-hour drive. I think most of you know you could never fly to Artsakh from Yerevan because Azerbaijan said they'll shoot down any airplanes. So you take the drive, and it's over the mountains. It's amazing. I think Salpy took this picture of me, and in the background is Mount Ararat, familiar to all Armenians, although it's actually in Turkey, I think. And it's supposedly the place where Noah's Ark ended up with all the animals when the floodwaters receded. Um, how he got the kangaroos to Australia, I'm not sure. That's never been explained. But anyway, these are nice myths. And I went to Stepanakert, which was, the main city there. This was taken just before we went into that main government building and met with the then president of the Artsakh region and got his buy-in to collect samples from his citizens there for genetic studies, which had never been done before. This is the study design of the work that was mentioned, funded at least a good portion of it by the Promise Armenian Institute. We had DNA we got from about 150 people. We made sure they were pure Artsakhis, at least up, through 3 generations. They didn't have any particular medical condition. And we did, this is what we could only do here, not there, what's called genotyping. It's a little like DNA sequencing, but you directly target certain letters of the genetic code throughout the genome. And as you can see, each of these people was tested for over 900,000 of these nucleotide sites. It's done on a high-density microarray, it's called. This is a picture of it. Another slang word for it is DNA chip, and it has about 1 million probes on it. Initially, it's huge data. We can mine it for the next 20 years probably. Initially, we chose to focus on only those mutations that were present at MAF. I don't want to get technical. It stands for minor allele frequency. If you multiply it approximately by 2, you get the percentage carrier rate. So we looked at any mutations that were in 1% or more of the population. We were especially interested in genes for hearing loss for obvious reasons because of Salpy's project, cardiovascular disease and cancer, the latter 2 because they're major killers in Armenia. So again, this refers to Salpy's cochlear implant project. In the middle with the scrubs is Akira Ishiyama, who's a wonderful guy who, as you heard, he goes there several times a year. Even as soon as the ceasefires are declared, he's off there to do more implants. Over the years, they've done around 250 of these. All of these children would have been deaf their entire life without this surgery. This costs like $40,000 in the West. If assuming you even have insurance, it wouldn't even be done. And they don't even know that surgery in Armenia, although Akira has been training the ENT surgeons there. What I didn't realize till Salpy told me is that none of these families have ever had genetic testing to see if there's a genetic cause for their child's hearing loss. That's standard of care in the United States. Any baby or older person diagnosed with hearing loss gets genetic testing. It helps to give genetic counseling for the recurrence risk if they have another child, for possible side effects, and so on. I had already had some experience with this because my own lab has done some screening for certain hearing loss mutations. 80% of deafness prelingual, that means before the baby has started to talk, it has a genetic basis. And usually it's what we call non-syndromic. They don't have any physical malformations or things like that. There's well over 100 genes that have been linked to it. The most common one is that GJB2, which is a recessive condition, but there are many, many others. We screened for that one in our local LA population. We had a grant from NIH to do that. And it was an ethnically diverse population, not Armenian. Babies, get a newborn hearing test. And if they fail it, it's not a great test because they're squirming around. So they have to fail it 3 consecutive times, and then they're referred to the genetics clinic where we would do the test. And we had a pretty high yield of that. But genetic deafness is very complex. This is a big, thick textbook I have in my office just dedicated to that. But as a first start, at least with the Artsakh data, we decided to look at some hearing loss genes. I know you can't read this, and it's just meant to show you the list of the variety of the wide nest we tried to cast. At the top, it's the GJB2 gene, and it has about the same carrier rate. It's about 2.5% that we find in the West. But the others there were 2 or 3 for a disorder called Usher syndrome, were much higher, 10 or even 100 times higher than the what we find in the European Genomic database, which is online to compare it to. And Armenians are basically Caucasians, so it's really the same racial group, but obviously quite different, maybe due to founder effect, as I said before. It's important to determine if the baby's hearing loss is due to Usher syndrome, because it also later in life causes retinitis pigmentosa, which is a cause of blindness. So it'd be very important to monitor for that. There's another one shown here, Pendred syndrome. Again, I think 100 times higher than in the regular European population. That one has thyroid problems with it. So it would, for all those reasons, I think it would be worth doing that kind of testing, and we're going to start working with Akira on that. Also, we looked at cardiovascular diseases. I'm not going to go into a bunch of details, but we found 3 genes with much higher carrier rate than in the Europeans that cause hereditary arrhythmias. Arrhythmia is an abnormal heart rhythm, which can be fatal. And the ones we found fall in the category of long QT syndromes. It's kind of a funny name. It has to do with what you observe in the EKG. The electrical impulses of the EKG are shown here. That's the normal pattern. The different troughs and peaks are labeled as P, Q, R, S, and T. The interval between the Q and T, which is shown at the bottom there, is what is prolonged in the long QT syndromes. And if it gets too long, the heart becomes unstable and can actually go into a much more serious, fatal type of arrhythmia called ventricular fibrillation. So these patients look fine, except they're at risk for sudden death. It's often by a trigger, a loud noise, some stress, sometimes diving into cold water, which if you knew they had this, you would advise them against that kind of thing. So that is one. I think it's dominant disease, but I think we could offer screening for it. And then the last one I just wanted to show is cancer. We did find a couple of mutations in the BRCA1 and 2 genes. I think you're familiar with those. They cause dominantly inherited breast ovarian cancer. But I'm not highlighting them here because the carrier rate is the same as in the European population, about 1 in 200. And at least now, for now, there is no recommended screening for BRCA mutations in the absence of a family history of those cancers. So I don't think we would recommend it. But we did find 3 genes, actually the 4 shown here, that are involved in dominantly inherited colon cancer, sometimes called Lynch syndrome. Again, just like with the hearing loss genes, some of these have other tumor risks like endometrial, small intestine, gastric. So it'd be very useful to identify the people at risk so they could get some scans at appropriate intervals and catch the cancer early. So just to return to these project goals, and I'll reemphasize the third one, which was technology transfer. We can't do this million probe DNA chip over in Armenia. They don't have that instrument. The chips in one run, they cost $25,000 just to do like 150 people. So you couldn't do it on millions of people. So somehow we have to adapt it to a cheaper technology. And our colleague Evgeny at University of Washington in his lab has already started to do that. He's put probes on these little strips, which we call dipsticks. It's kind of like to do a crude urinalysis. You dip a thing in, it changes color, or like a pregnancy test. So, but each of these bands is a certain mutation. It's called multiplex because he was able to put the mutations for 4 or 5 or 6 genes all on one strip. This could probably be done for, I don't know, $1 each or something. You know, it's really much cheaper. So our project is the discovery phase. We're going to look for 1 million different mutations, but we'll narrow it down to the most significant ones to then be put on a much less expensive platform. Again, I have some experience with this. I had a grant in the 90s from NIH to check the feasibility of screening for the most common recessive disease in North America, which is cystic fibrosis. And it worked very well. It ended up to be a, excuse me, a national recommendation that all pregnant couples must be offered CF carrier screening. And so this involves tens of millions of people getting tested. But we ended up using, we pioneered it actually, the same kind of strips with the mutant probes already on them. In this case, there were 25 mutations, all for that one gene. So there is a precedent for doing that on large populations. In fact, the test my lab uses for the FMF mutations also uses this strip, although it's commercially made, a company in Austria. So our findings, keep in mind, they're somewhat preliminary. They need to be checked on larger numbers of people. But if validated, I think it could help inform us to introduce in Armenia and maybe in the diaspora public health programs customized for the Armenian population. This could be carrier screening for recessive disorders that would affect the child, prenatal diagnosis, newborn screening, or population screening for some of the adult cancers we talked about. Most of this is not being done at all in Armenia. Newborn screening in the US, in all 50 states now, they screen for at least 50 recessive diseases. I think in Armenia it's only 1 or 2, PKU being the most common one. So any of this would be an advance to what's being done now. These are the limitations. You know, 150 is a small cohort. We do want to confirm it. Some of the variants we found, we're not totally sure yet how disease-causing they are, so that needs to be looked at. And most important, does the Artsakh data extend to the general Armenian population over there? And if it does, of course, it would be over here as well. I was hoping I'd have some results to show you that tonight. I don't. It didn't quite come in. Our gut feeling is that it will be similar. Although Artsakh is very isolated, it's a reason we chose to use it because it's the most isolated region in Armenia, and geneticists like to have pure populations like that. But even though it's 5,000 or 8,000 years old, in the time scale of human evolution, that's like a microsecond. So I don't think it's had time to differ that much from the general Armenian population. We also have some other results. This was done long before the PAI project with a genetic counselor, Ida, who's now working at one of our community hospitals. She took one of the commercial carrier screening platforms that pregnant couples are offered now, 176 different conditions, and kind of mined the genome sequences we had already done on about 50 general Armenians. There were a few Artsakh people in there as well. And the frequencies she got do seem to resemble what we're seeing in the Artsakh. I haven't done this in detail yet, but I've circled the gene for cystic fibrosis, the genes for PKU, phenylketonuria, which is very common. And of course, I haven't mentioned it, but in all of these studies, we find the expected 20% carrier rate for the FMF mutations. So that kind of goes without saying. So a little bit of acknowledgments. I've already, the various, I and the introducers, the moderators have already mentioned a number of these at UCLA, in Armenia, and the various grants we've had. We especially want to thank the PAI for that. And I just wanted to end with this. I've showed you a couple times the project goals, but what was never listed as a goal, but turned out to be, I think, a valuable outcome of this. In choosing to study the Artsakh people, we've kind of kept them visible, with what's going on in Ukraine and Israel, Palestine. Nobody's paying any attention to Armenia and Artsakh. You all know that. And I think anything we can do to raise awareness of the humanitarian crisis, they had a blockade and all that, is of help. Also, this is a great credit to the Artsakh people, even in the midst of all their suffering, even the ones in the burn unit that Salpi mentioned. When you ask them if they want to be part of this project, they're very eager to do it, even though this must be like 50th on their list of priorities right now. They're just trying to find a place to live. So to the extent that that can keep going, I think that's a valuable offshoot of this project. Whatever we do, we're going to do it very carefully. Everything will be fully validated and checked with the community and so on before it's rolled out. That's not always done in the United States, and we don't want to fall victim to this kind of thing where there's a lot of direct-to-consumer genetic testing going on, really without any genetic counseling involved or anything like that. So I'll stop there. Thanks again to the organizers for inviting me, and we'll have plenty of time for questions at the end, I think. Yeah. So I'm introducing, oh, yeah, go ahead. Yeah, who am I introducing now? Okay. Yeah, thank you. Thanks for coming.
Hello, I'm Evgeny Sekurenko. Thank you very much for organizing this. I feel intimidated by it. So, thank you so much. It's a wonderful event and it's a wonderful work, Dr. Grody. Thank you so much, Promise Institute. You can tell by my accent, I'm not Armenian. Yeah, and I actually in this country almost 25 years, and I have to practice this accent every day to keep it still strong. Yeah, so. And I am, as Dr. Grody said, he considers himself ABC Armenian by choice. I'm not Armenian by choice. My choice was to marry Armenian. And then I had no choice but to become Armenian, which I'm very happy and proud of. I'm Ukrainian, yeah, I'm from Ukraine, and actually there is a very deep connection between Armenia and Ukraine. Ukraine has one of the largest diasporas, Armenians, and very, very ancient Armenian diaspora there. And you know what? So this is Artsakh flag, but this is Armenian flag, yes? This is Ukrainian flag. Armenian, Ukrainian. There is no closer flag to Ukrainian one than Armenian. And the thing is, what is why it's red? In 2020, when that happened with Azerbaijan occupying Karabakh, it gave Ukraine a very important lesson. Don't trust your enemies that they will stay away from you, and get really good weapons to protect yourself. Okay, so Armenians paid with their blood for this flag still to exist. So thank you very much actually for that. Yeah, it's very, very important for us. So again, I'm, as I said, I'm a microbiologist. And you wonder, what I have to do with this human genetics project. My wife, Arpenik Avakian, sorry, forgot the name to mention, and she's a ophthalmologist. So I actually microbiologist, but you know what we say in microbiology, what is true for E. coli is true for elephant, only more so. So, and humans are they're not as evolved actually, and not as kind of elephants are, but still, so we can put the connections. So, just saying that, genetics is actually very close to me, and I always was fascinated with any type of genetics, human genetics too. And let me emphasize why we started this project with Armenia. Not only because, I had no choice to become Armenian, and the Armenians are close to me in many respects. Armenians is very cohesive nation, not very big nation, that didn't have so-called admixture, genetic admixture, so didn't have too many blood from other nations to come and mix. This creates a fantastic possibility to find very important genetic changes due to what Dr. Grody said, founders effects and so on. So it's very, very useful. And here we have Dr. Jordan, who started the Armenian bone marrow registry, who can tell you that Armenians match each other much more often in bone marrow transplants, and so on. And that's what it is. Genetic cohesiveness is similarity is very, very important to find different types of mutations at higher rate, as you had seen. So purely from scientific point, this actually made sense. No one had done this before. Karabakh people are even more homogeneous part of than the entire Armenians, because they were not displaced for thousands of years from their region. This is the most ancient part of Armenians that didn't have the admixture of other genes in it. That's why studying them, even of a bigger value to find different types of genetic changes that would be harder to find in general Armenian population. What is also very critical, and Dr. Grody mentions, is that sometimes we see so often some kind of genetic changes that we think that there is some kind of selective advantage, some of these pathogenic mutations that are bad, there is too many of them. And actually, you remember when we were looking at the data, everyone in the Linguist Institute were wondering how these people are surviving. How are they still alive? Such a huge frequency of these bad mutations. And this actually tells us that these mutations have some other reason to exist. Or there is mutations that give selective advantage for these people, or there is other mutations that compensating them. Finding these mutations that neutralizing the effect of bad mutations will be of absolutely huge global importance, because that's how we will understand how to fight many diseases, what compensate for them. This maybe I'm trying to be too technical, but this is of absolutely unprecedented importance for us to really study this in depth. This could have not only for the Karabakh, Artsakh people, not only for the Armenians. This could be an absolutely importance for the us as a humanity to study in depth specifically these populations. So I really, really want to emphasize how important for us to keep doing this. And I really hope that my enthusiasm somehow translates to yours. So thank you very much. And by the way, I wouldn't say that this country doesn't exist anymore. There is a history is, playing. So we keep fighting, and we will stay together. Good people stay together, and we will win. Thank you so much.
Okay. It's going to be hard for me to continue after these lovely presentations. Thank you. Ladies and gentlemen, dear colleagues and friends, let me first extend heartfelt gratitude to UCLA Promise Institute for organizing this important meeting. I'm honored to be here today and to have the opportunity to share with our vision of Armenia, the Armenian Genome Project, launched by a group of enthusiasts back in 2015, the centennial of Armenian genocide. As you understood, that among the enthusiasts was Dr. Yevgeny Sakuranko, who has taken the key role of the idea generator and promoter. Well, there are a couple of reasons why he undertook this role, but we all know one of the important one. The project is therefore designed to explore and highlight the historical pathways leading to our origins. Those are the general goals and ideas of the project. Construct the living map of Armenian based on genetic makeup of people. Establish a national gene bank consisting of phenotype and genotype data of the Armenian population. Define specific inherited traits and deepen the understanding of the genetic roots of the Armenians. Use genetic technology to identify patients with gene variations that raises their risk of certain diseases. And enforce, of course, the development of genomics research in Armenia, participation of Armenian researchers in international genome projects. Well, here are some timelines of our activities represented by the following events and operations. A group of Armenian researchers launched the program, as I mentioned earlier. And I have to mention that this initiative was made possible with seed funding generously donated by late Ms. Sarah Chijan. In 2016, an MOU was signed between UCLA and the Armenia to begin a data collection, and Dr. Grody was assigned as a principal investigator of UCLA Armenia project, and we're lucky and happy to have Dr. Wayne as our team member, and we thank him for his time, generous time spending with us and sharing his knowledge. And a few years later, in 2021, recognizing the value of UCLA Armenia project, the UCLA Promise Armenia Institute granted funds to Dr. Grody to continue the study. And I'm going to thank Anne Garagozian. She's been very helpful to overcome all the difficulties and bureaucratic obstacles of UCLA. Dr. Salpe Agarakian was very instrumental in bridging not only our project with UCLA, but also during all these years, the registered nurse program in Armenia and UCLA, and of course the cochlear implant project. And I assure you that Dr. Ishiyama is a very popular, not the most popular doctor in Armenia. An MOU in 2017 was signed in between Armenia and the Ministry of Health of Karabakh. And this gave us the opportunity to collect blood samples from Artsakh back that time. And by the way, Ara Harutyunyan, who was at that time the prime minister, was the godfather of our project and agreement. But as you might know, that unfortunately he is now in jail in Baku. I'm going to stop here, but I assure you that this is the least that we can do for people from Karabakh. And I think that doing this research amongst them will bring us to another level of providing healthcare to those who suffered lately. I'm talking about the personalized medicine. We're talking about medicine that is based on genetic analysis. And this is another level even for Armenians living in Armenia. And let's do it for them. And we don't have much time because, as Eugene mentioned earlier, these people will, assimilate with the others. They will lose their the signal of their genes coming from thousands of years due to isolation. and after 1 or 2 generations will be the same. They will be the same as we are. So this project intends to start this approach, and I hope that we will have a big army of enthusiasts to join us and to help these people. And that's it. I'm done. Thank you. We're ready to answer to all the questions.
Thank you so much to Wayne and Yevgeny and Sevag, Wayne especially, for just a magnificent overview and technical presentation, but with very important applications. And I'm very glad that, Sevag, you mentioned the individuals who are Artsakhs who are still being held captive by the Azeri government. Many of the leaders that you perhaps engaged in order to acquire these samples are now being held. And so I hope we will not forget these individuals, keep them in our hearts, in our prayers, and agitate for their release. So we thank you for reminding us. So now I would like to open the floor to questions from our audience. We have Emily Pogosyan here who can come to you with the microphone if you'd like to order them, offer them. So we have, why don't you go ahead and offer them, and then I'll give this to the person who is being questioned.
Thank you. Thank you very much for your presentations. Regarding the Mediterranean fever disease, are there any environmental factor that triggers the expression of the gene? Have you checked from epigenetics perspective?
That's a wonderful question. The answer is we do suspect that. We haven't looked at the epigenetics for those not in genetics. Epigenetics means a change in gene expression not due to a change in the sequence of the DNA. It's some higher level regulation. We do see the risk of complications of FMF differ by geographic origin. We've never been able to explain it. So patients with the same mutations, if they come from like Sephardic Jews from North Africa, they have a very high risk of the complication of amyloidosis. That's the one life-threatening aspect of FMF. Whereas Ashkenazi Jews with the same mutations from Eastern Europe don't have any problem, even if they're not treated. I mean, they should still be treated. In the days before Colchicine, people would self-treat as empirically, and many people found if they avoided spicy foods or fatty foods, it would decrease the frequency of attacks. Luckily, now if you take your colchicine, there's no dietary restrictions at all. We do know that stress can trigger an attack, even in people who are well controlled with colchicine, and that's not always avoidable. So there are definitely environmental issues. The high-risk people, even Yerevan is considered high risk for amyloid. For some reason, when they're in this country for a couple generations, that risk goes down. So that must be something epigenetic due to diet or weather or something. We just don't know.
Thank you.
Hi, thank you so much. First of all, I want to thank the organizers for putting together such an amazing set of presentations, and particularly our presenters, Dr. Grody. It's good to be back on my old stomping grounds. I used to be a student here over 20 years ago, I think, if my math is correct, and actually was a genetics major, and worked in Jake Lucis's lab, who was also married to an Armenian.
Yeah, I knew that.
There he is. Hi, Mark. I didn't see you there. Since then, I've gone onto the clinical side, and though I don't do genetics myself, I'm always interested in how genetics interfaces with, all of our clinical practices. So one of the slides you presented was really about the future and what the next steps are. and you had envisioned genetic screening for various populations. So my question is, are you suggesting we have a different genetic screen for the Armenian population versus the standard Californian population, for example? Or how do you see that working out in the long run?
Yeah, that's a great question. I don't think we have those answers yet exactly. You have to go a little bit into the history of genetic screening. And a lot of this dates before the era of next generation sequencing, where we could look at all the genes for not that much money. In the old days, it was really expensive. So you had to pick and choose the genes that were the highest frequency in that ethnic group. So Italians would just be checked for thalassemia, for example, because they have a high carrier rate, even though you can find thalassemia in every other group as well. Nowadays in the United States, using next generation sequencing, like I said, there are commercial companies that test for 300 diseases. I don't necessarily approve of that. Most of them I've never heard of. I don't think we know their natural history or anything. That's not ready, to my knowledge, to be done in Armenia yet. So if we could target the highest frequency mutations that we find there in a inexpensive way, like those dipsticks that Evgeny's been working on, that could be a rationale for customizing the screening for that group. It might be different in the homeland versus here, where I think anyone with health insurance is going to get the 300 diseases anyway. I think much of this may be a moot point moving forward. Probably within the next 5 to 10 years, every newborn's going to get a whole genome screen, probably whether they like it or not. I don't know what will be done with the information. Might be stored in the electronic medical record for some future use. I have major ethical problems with that. But if that does come to pass, then everyone will have been screened from the time of birth for all 22,000 genes. So this is really a moving target. But I think what we're interested in now is developing something. There's so little screening going on in Armenia now. I think anything we come up with that targets, is customized for that group and not expensive would be a help, at least in the intermediate term.
I think we have a question right here.
Thank you. First and foremost, I'd like to thank you for the work you guys have been doing. I am a first-generation descendant of 2 Artsakh refugees, so I specifically thank you for this. And it's very personal to me. So I was wondering, while getting your samples, were you focused on some regions more than others, or did you get a broad sample size from across all regions of Artsakh?
I'll turn this over to our experts.
When we signed the agreement with the Ministry of Health of Artsakh, the target was 2,000 people, and we just collected random samples. And I'm sure we were focused on Stepanakert, where at the hospital where we were visiting, we have representatives of all 5-6 regions of Artsakh. So we didn't target specifically this or the other region, but it was a random pickup of our patients and blood samples. So if that answers your question. Sure.
If I may add to Sevak's comment, the last batch of samples that we were hoping the results will be ready for us today, they're not. They are from the burn patients, and we had about 50 samples from the burn patients. And when you look at their demographics, they're from different villages. So they weren't all from Stepanakert. So we are tracking that data. We do have the data exactly what part of Artsakh these samples are coming from, which will be an analysis later on. But right now we're just looking as a whole Artsakh. Okay, thank you.
So I think we have a question right here.
First of all, thank you so much, all 3 of you, for the wonderful presentation. My question is for Dr. Grody. Do you know how far back in time we can trace the FMF mutation? I'm an archaeologist, so that was the first question that popped in mind. And have you looked at ancient Armenian DNA work that has been done in the last couple of decades to see if the mutation shows up in any of the sequencing they have done?
Yeah, that's another great question. I can only quote from papers in the literature by other people, and I don't know how much was done by directly sequencing so-called ancient DNA versus more your field, just monitoring the patterns of migration. That all the papers I've read say that these are very ancient mutations. They long predate the birth of Judaism, let alone Christianity, Islam. I mean, they may go back 8,000 years. The reason that's an important question is what we talked about at the beginning. Is this a founder effect, or does it really have a selective advantage? Evgeny, his bias is that why would it be there if it didn't have an advantage? And he may be right. As best we can tell, though, FMF patients and their carrier parents, their immune systems are no better and no worse than anyone else. And that came up during the COVID thing. The parents would ask me, is he more susceptible to COVID? I said, no, but get the vaccine anyway. And, so whatever it is, what I mentioned about the Black Plague bacillus, that's only been done in mouse models. So I don't know how it extends to humans. So it's really people in your field who study ancient human migrations who can best answer that.
Could I ask a quick question kind of related to this? So the MEFV gene, of which there are dozens of mutations associated with the FMF disease, do Armenians tend to have certain mutations that are different from other ethnic groups or similar to a couple of different ethnic groups? Maybe you could comment on that.
Yeah, that we have a lot of direct experience, plus in the literature. You heard that our clinic is the biggest in the Western Hemisphere, but it's dwarfed by what's in Yerevan, and also there's one in Ankara. So, that's where the bulk of the patients are. So there is a lot of information on this. Pretty much whatever patients I see and do the DNA test, I see the same. There's 4 common mutations that I see over and over. And it doesn't really matter if they're Armenian, Arab, Jewish, or as I mentioned, even Japanese and Italian. Our own test looks for the 12 most common mutations. As Anne mentioned, I think there's now 200 cataloged. Most of the others are very rare. I've found that if my 12 mutation test doesn't pick it up, even sequencing the whole gene doesn't find it either. So it must be something very unusual. So again, it gets back to your question, where did these things start? I'm pretty sure they started in the Fertile Crescent and then went throughout Europe, Africa, and then somehow to Asia.
Hi. I have a two-pronged question. One regarding FMF. Since you know so much about the exact loci where the mutations are attributed to the disease, is there any chance, in your opinion, of CRISPR, applying CRISPR to maybe, and I'm not talking immediately right now, but the future? And then my second question is about the difference between Tay-Sachs versus FMF. From what I understand, Tay-Sachs, again, I could be completely wrong, but what I read about, because of the persecution of the Jewish people, they were trying to interbreed and keep their communities or wealth at one place. Is that a reason that Tay-Sachs or the Fanconi or other Jewish panel diseases are different than FMF? Because FMF looks to me like it's just a mutation that wasn't caused necessarily by a specific ethnic group.
Yeah, I guess because it is in different ethnic group. Now, keep in mind, whenever we talk about ethnic risks for certain genetic diseases, we talk about the groups that have the highest carrier rate. It doesn't mean it is not found in others. You can find sickle cell in non-Africans. But the two stories may be different. I think what you were hinting at with Tay-Sachs, I'm pretty convinced it's a founder effect. There's about 10 or anywhere to 30 so-called Jewish recessive diseases. I think they're all founder effects. And it wasn't just the initial founding of the population, whenever that was, 6,000 years ago, but there's been bottlenecks. And this is where the two groups have things in common, namely genocides.
Yeah.
So you may start out with a quite diverse population, and then it gets narrowed down to a fairly uniform population just by bad luck. Again, it doesn't answer the question of selective advantage, but it's just the FMF one is such high frequency that there's no other example of that anywhere in the world. So now your first question about CRISPR, just so people understand, that's the one of the ways to do gene editing, which is the thing that has kind of replaced gene replacement therapy. And it's far more easier to do and cheaper and more successful. There was a Nobel Prize given for it in 2020. So he's asking, could we use gene editing in an FMF patient to remove the mutation, restore the gene back to its normal sequence? That would be the goal for any disease. There isn't any research I'm aware of to do that in FMF because it's considered so easily treatable with colchicine, which is fairly cheap to begin with. They're really looking at the much more fatal, serious diseases and cancer for CRISPR. Yeah.
Thank you.
Hi, thank you so much for your presentation. It really hit home for me because myself and my siblings, we were born in Armenia, and two of my siblings have PKU. And as I know, in Armenia, the screening for PKU started in 2008. My little brother, he was born in 2014, and he was not screened for it, and he was left undiagnosed for 8.5 years until we moved to America. And where my little sister was born, in 2022. Oh my God, she's so little. And yeah, she was diagnosed with PKU, and then they realized that my little brother had it. So I just wanted to emphasize how important genetic screening is in Armenia, because while they say that they have it, and statistically, yeah, they have it, some people, they find it, because as you mentioned, there's 50 different screenings here and only one or two there. But again, these one or two, they're not always carried out. So I just wanted to emphasize how important this is, because my brother was left undiagnosed for 8.5 years, which is crazy for PKU. But we are so lucky that we found it before he turned 10 years old and they started treatment for him. But yeah, it's just really important, and just being here makes me so happy that just in the future, cases like my family's wouldn't happen. And I think I'm the youngest one in the audience here. I'm a freshman in high school, so I'm nowhere near college level yet, but I am gonna go into genetics, and this is just an amazing experience, and thank you so much.
Oh, thank you for sharing that. Great. Yeah, that's the story right there. And I knew PKU came late to Armenia, the screening. It was the first newborn screen in the US. It actually started in the 60s. Robert Guthrie, who had, I think, a nephew with PKU, developed the assay for it. And then they added galactosemia and a few other things. Now it's up to 50, like I said, but really only in the West. And that's an example like FMF. If you identify the babies right away, the treatment is actually dietary. The disease is phenylketonuria. They cannot metabolize the amino acid phenylalanine, which is in all the protein we eat, meat and everything else. And so you just put them on a low phenylalanine formula. It's called low phenylac. And if you don't, toxins build up in the body that are mainly, as you know, injurious to the brain. So intellectual disability, what we used to call mental retardation, is really the symptom. And in fact, there is no other physical symptom. That's why they go undiagnosed for so long. That shouldn't happen, although we've had, even in our clinic, we've had some children who are missed. And it's awful. Once the brain is damaged, that's it. Nothing much you can do. I'll be happy to meet with you for career counseling if you want. I can't guarantee getting into UCLA. My own son didn't get in here, so.
Do you have a comment?
I want to come back to Dr. Garni Barkhadurian's question. Dr. Garni Barkhadurian, first of all, is the president of Armenian American Medical Society, which Anne mentioned, they're one of our sponsors. But coming back to the screening, when you look up the MOHs, and Sebag, you have to help me on this. When you look up at the MOH's website, there are only 6 different newborn screening in Armenia. And I was going to say PKU, one of them, but now we just heard an eyewitness story that even though the screening is mandatory, is it really happening? It looks like it's not 100%. Okay. So PKU is one of them, then hypothyroidism in Armenia for the newborn screening, hearing loss, which interestingly in the United States, California mandates, and Dr. Isham has been pushing to get other states to mandate, and I think few more are mandating. But Armenia is doing hearing loss, and that's because our experience over there. Critical congenital heart defects. This is something that in the states in California, not too long ago, in the nurseries, we started screening for the congenital heart defects. And it's an easy, obviously, screening. You just put a pulse ox on the kid's finger and you can figure out if they're oxygenating adequately or not. And then eyesight for low weight or premature births, which again, Dr. Roger Ohanessian's group pushed this through the MOH to get this screening going. And then hip dislocation, which something that it's done actually when you go to the doctor's office. But I want to turn it back to Sebag about something that when we were in Armenia, we met with Deputy Minister of Nanushian as a future step, because we're talking about, okay, so what are we doing next? The analysis is going to continue. We've got lots of samples getting ready to be analyzed, and we'll get the reports. But then what else is going on? So when we met with Dr. Nanushian, we talked about how do we translate this research information to clinical settings, and can we validate it, what's going on clinically? And so there was, you tell the story. Do you want to talk about, okay. So we wanted to then go back to see in the clinic settings, how many of our patients that we said genetically have cardiac arrest from prolonged QT interval, how many of our patients clinically actually in the hospital setting had that, and does it really then match with our research findings? So there are a few of those findings we're trying to also validate it clinically. So there's a lot to be done in Armenia. We're just beginning, actually, even though it's been going on since 2016, but there's a lot we have to continue discovering, implementing, and doing the screening a little bit better and increase the screening. So I just wanted to mention that Armenia's got a long way to go. When you look at the US screening, this is all the things that Dr. Grody said, how many genes, but this is the list that is required compared to Armenia, only this. So, but we'll get there. We'll get there.
Great, thank you. Maybe Christine, one more question?
I do have one more question about, this is not necessarily genetic screening, but screening mothers, expecting mothers who are of 35 years old plus, because it's done here in the US for certain screening, I guess, the fetus for certain genetic mutations. Is this a thing in Armenia? Do they do it? Or is this something that you have ever considered getting started there? Because that's also important for a number of different reasons.
Yeah, I'll just define what she's referring to and then turn it over to my Armenian colleagues. She's talking about NIPS, non-invasive prenatal screening, which started, I don't know, around 10 years ago. Instead of doing an amniocentesis, which, you know, has a risk of causing a miscarriage when you stick the needle in, you just draw peripheral blood from the pregnant mother. quite fairly early in the pregnancy, and through next generation DNA sequencing, you can detect chromosome problems like Down syndrome, Turner syndrome, and so on. Usually it would need to be confirmed by an amnio later, but it saves money and it saves risk to a lot of babies. So that's now offered to all pregnant women. They used to only offer it to the ones who were offered amnio, which was over age 35. Now they've just decided to offer it to everyone. I have no idea if that's done in Armenia. Do you guys? Okay, he says no. This would be an obstetric issue. And again, it requires the fancy next generation DNA sequencers, which cost about $1 million each. So that may be the limitation.
All right. Well, I think if there are no further questions, we can thank once again our magnificent speakers, Dr. Wayne Grody, Dr. Seva Avakian, Dr. Yevgeny Sarchenko. Sokuria. Oh, okay. And thank you so much. This was just a magnificent series of presentations. Before we end, let me just alert you to the fact that our Promise Armenian Institute has a number of upcoming events. I'll just tell you about two of them. One is taking place on Saturday, February 10 at 10 a.m. Pacific. This will be in the Fowler Museum, north part of our campus. And this will be an all-day conference that is sponsored by our UCLA Armenian Genocide Research Program. Dr. Taner Akçam, the director, is sitting in our audience. And the subject is Armenian Genocide, Looted Art and Restitution. This is a follow-on conference to the conference we held a little less than a year ago on genocide recognition and restitution in the post-recognition era in the United States. This will be an open event to our public, so feel free to come to our Promise Armenian Institute website. It will also be live streamed on Zoom and on YouTube. And then also as a heads up, on Saturday, April 6th, the UCLA Promise Armenian Institute and partners are in the process of planning an all-day symposium in memory of our beloved late Professor Richard Hovhannisyan, who for more than 50 years served on our faculty in Armenian history, was a very distinguished scholar, and enhanced our understanding of Armenian history enormously worldwide. And so that will be in this room, Saturday, April 6th. Please, again, consult our website and social media to learn more about this. So once again, thank you so much for attending. I'd like to thank our very hardworking staff in the Promise Armenian Institute, Hasmik Bagdasarian, Emily Pogosyan, Nanar Hartunian, and thanks to all of you for being here. I'd now invite you to join us for refreshments in the lobby. So thank you again.